morning, students. Today we will continue with our chapter number 2, the reproduction in plants. We have already completed the vegetative reproduction. Today we will study about the tissue culture method. Tissue culture is a technique of raising the new plants from a small portion of the plant tissue over a culture or any synthetic medium under the sterilized condition, that is the germ-free conditions are created. For plant, the synthetic medium plays very important role as it contains various nutrients and plant hormones, that is the chemicals that help in the growth of the plants. Now, what is the method of tissue culture? The steps which are followed in tissue culture are as follows. We take a small piece of plant tissue from the growing tips of plant, such as tip of root or shoot. The tissue is transferred in a test tube containing a suitable nutrient medium under the sterile conditions. This medium contains plant hormones. Due to the presence of plant hormones, the tissue divides and forms an undifferentiated mass of cells called callus. The callus differentiates into plantlets. These plantlets are transferred into pots or soil to attain maturity. By this technique, we obtain disease-free plants from any diseased plant, as plants propagate from shoot tips, which are often disease-free. Advantages of tissue culture. It allows the rapid multiplication. That is, a large number of plants can be developed in a short period of time. Little space is needed for growing the tissue culture. The propagation of tissue culture can be carried out throughout the year, irrespective of seasons. The technique of tissue culture is used for production of ornamental plants like orchids, dahlia, carnations, etc. Now, the sexual reproduction. Sexual reproduction mostly takes place in flowering plants and in some non-flowering plants. The flower is the most attractive and the colorful part of the plant. It bears both the male reproductive organ and the female reproductive organ that contain the male gametes called pollen and the female gametes called egg or ovum. A typical flower consists of 4 whorls: the calyx or sepals, the corolla or the petals, androecium, that is the male part, and the gynoecium, which is the female part of the plant. All the 4 whorls are arranged on a flattened tip called thalamus. The thalamus is attached to the main stem by means of a stalk called pedicel. Calyx is the outermost whorl, which contains green leaf-like structures called the sepals. The sepals protect the flower at its bud stage. The second whorl consists of the colorful petals. The petals are often large and scented. They produce the nectar. Androecium constitutes the male reproductive part of the flower. The androecium consists of stamens. Each stamen has 2 parts, an anther and the filament. Anther bears numerous pollen grains which produce the male gametes. Filament is thread-like structure which holds the anther. Gynoecium constitutes the female reproductive part. It consists of the pistil or carpel. Each pistil consists of 3 parts, namely stigma, style and ovary. The stigma is a sticky structure at the top of the pistil that receives the pollen grain. The style is the stalk of the pistil. The swollen base of the pistil is called ovary. The ovary contain ovules. Each ovule consists of an egg or a female gamete. The first two whorls, that is calyx and corolla, do not take part in reproduction and are known as accessory whorls. The inner two whorls, that is androecium and gynoecium, are directly concerned with the sexual reproduction, and so they are called essential whorls. If a flower has got all the four whorls, that is calyx, corolla, androecium and gynoecium, it is called a complete flower or a bisexual flower. In the incomplete flower, either androecium or gynoecium is not present. That is why they are also called the unisexual plants. Differences between asexual reproduction and sexual reproduction. In asexual reproduction, only one parent, either male or female, is required. In sexual reproduction, both the parents are required. Asexual reproduction, the new plants are identical to the parent plant. In the sexual reproduction, the newly developed plant is not the same as the parent plant. Asexual reproduction occurs in the lower organisms through fission, budding, fragmentation, sporulation, and vegetative propagation. Sexual reproduction requires fertilization, in which fusion of male and female gametes takes place. Pollination. Pollination is the transfer of pollen grains from anther to the stigma of a pistil. All the flowering plants are amazingly adapted to achieve the pollination. Pollination is of two types: self-pollination and cross-pollination. Self-pollination. In this, the transfer of the pollen grains from the anther to the stigma of the same flower or another flower of the same plant takes place. Cross-pollination. In cross-pollination, the transfer of pollen grains from the anther of one flower to the stigma of another flower of a different plant of the same variety takes place. For cross-pollination, agents like wind, water, and insects play an important role in transferring the pollen grains from anther to the stigma of another flower. So they are also called the pollinators. Cross-pollination by insects. Insects like bees, butterfly, moths, etc. visit the flowers to get the nectar found at the base of the petals. While they are feeding, they accidentally rub against the stamens and pollen grains stuck all over their body. When they visit another flower, some of the pollen grains are transferred to the stigma of that flower. Examples: rose, marigold, and jasmine. The cross-pollination by the help of wind. Some flowers like rice, maize, grass, etc., when they get matured, the pollen grains are blown away by the wind. Some of these pollen fall on the stigma of another flower, resulting in pollination. Pollination by means of water. In the aquatic plants, the male flowers are completely submerged in water. On maturity, they get detached and float on the surface of water. When they come in contact with the female flower, the pollen grains fall on the stigma, thus leading to pollination. Characteristics of insect pollinated plants. They are large in size, they are brightly colored and sweet smelling flowers. They produce nectar in a large quantity. Pollinated flowers are small in size. They have dry and light pollen grains which can be easily carried away by the wind. The pollen grains are produced in large quantities so that at least some get chance to reach the stigma. The style of female flower is projected out and has a large feathery stigma for receiving the flying pollen in the wind. The flowers of water pollinated plants are very light so that they can easily float in water. They do not produce nectar. They are produced in large quantity. The pollen grains are produced in large quantity, and the male and female flowers are produced on separate plants. Artificial pollination. In the artificial pollination, the stamens of a bisexual flower, that is, the flowers having gynoecium and androecium both, are removed before their anthers mature. This is called emasculation. The flower with gynoecium is then covered with a muslin bag to prevent the natural pollination. This is called bagging. When the stigma of the flower gets matured, pollen from another selected plant is taken and deposited on the stigma. Thus, the artificial pollination takes place. The seeds produced after artificial pollination give rise to a new hybrid variety which is high yielding and disease resistant. This process is also called hybridization. Fertilization. Fertilization is the process of fusion of male cells, that is pollen in case of plants, with the female cells or egg cell to form the single cell which is called the zygote. The zygote divides and redivides, growing to form an embryo and then a seed. The seed then grows into a new. The process of fertilization takes place in the following steps. The pollen grains fall on the stigma, and by absorbing water and nutrients from the stigma, they germinate to produce two nucleus, the tube nucleus and the germinative nucleus. The tube nucleus forms the pollen tube, and germinative nucleus further divides and forms two sperm nuclei. The pollen tube grows through the style and finally reaches the ovule of the ovary through the micropyle. One of the two male nuclei present in the pollen tube fuses with the egg cell and forms a zygote, which develops into an embryo. The other male nuclei fuses with the polar nuclei of the ovule to form endosperm, which nourishes the developed embryo. The differences between pollination and fertilization. Pollination is a physical process, while fertilization is a physiochemical process. Pollination is the transfer of pollen grains from anther to the stigma of a flower. Fertilization is fusion of male and female gametes. In pollination, external agents may be required to complete the process. Fertilization is an internal process which takes place within the ovule of the ovary. Thank you.